If we take a cross-section of the kidney, there are two main parts, the outer cortex and the inner medulla. If we zoom in, there are millions of tiny functional units called nephrons, which go from the outer cortex down into the medulla and back out into the cortex again. These nephrons perform the major function of the kidney, which is to clear harmful substances from the body by filtering the blood. And each nephron has a glomerulus, or tiny clump of capillaries where blood filtration starts. Interestingly, once the blood leaves these glomeruli, it doesn't enter the venules. Instead, the glomerulus funnels blood into the efferent arterioles, which divide into capillaries a second time. These paratubular capillaries then reunite, and at that point the blood enters venules, and eventually drains back into the venous system. Now, when blood gets filtered, some fluid stays in the glomerulus, and some fluid goes into the renal tubule. The renal tubule is a structure with several segments: the proximal convoluted tubule, the U-shaped loop of Henle, with a descending and ascending limb, and the distal convoluted tubule, which winds and twists back up again, before emptying into the collecting duct, which collects the final urine. Now, zooming in on this nephron's tubule, each one's lined by brush border cells which have two surfaces. One is the apical surface that faces the tubular lumen, and is lined with microvilli, which are tiny little projections that increase the cell's surface area to help with solute reabsorption. The other is the basolateral surface, which faces the paratubular capillaries, which run alongside the nephron. Alright, so one substance that's filtered out of the glomerulus and into the tubule is para-aminohippuric acid, or PAH for short. Actually, para-aminohippuric acid is an organic acid, and about 90% is bound to plasma proteins. So really only the unbound 10% can pass through the glomerular capillaries. Essentially, the higher the unbound PAH concentration, the more PAH will get filtered. If we wanted to illustrate this in a graph, with the unbound PAH concentration in the blood on the x-axis, and the PAH filtered on the y-axis, we'd see that as the unbound concentration of PAH increases, the filtered load of it also increases in a linear fashion. Now, in the renal tubule, reabsorption is when something moves from the renal tubule back into the blood, and actually no PAH is reabsorbed. Secretion, on the other hand, is when something from the paratubular capillaries moves into the renal tubule. And in fact, an additional bit of PAH is secreted from the paratubular capillary blood into tubular fluid. To accomplish this, the basolateral membrane of the tubular cells has a special type of carrier protein that transports PAH, as well as other organic anions. So let's add that PAH that gets secreted to the graph, also on the y-axis. At first, the higher the PAH concentration in the blood, the more PAH will get secreted into the tubule. But there's only a limited number of carrier proteins available on those tubular cells, which means that at some point they're working at full capacity, and no additional PAH will allow them to work any faster. This is known as the TM, or transport maximum. After this point, the secretion of PAH stays constant, even with increasing PAH concentration. Finally, we can add the PAH that gets excreted in the urine to the graph. And excretion in the urine is the sum of the filtered PAH plus the secreted PAH. In our graph, below the transport maximum, small increases in plasma PAH concentration gives rise to steep increases in the PAH that gets excreted, since both filtration and secretion are rising. Above the transport maximum, though, secretion gets saturated. So with increases in plasma PAH concentration, only the filtration component increases, and excretion rises less sharply and becomes parallel to the filtration curve. Alright, so the reason that PAH matters is that it can be used to estimate the blood plasma reaching the kidneys per unit time. Just like any other substance that reaches the kidneys, the amount of PAH entering the kidney has to equal the amount of PAH leaving the kidney, and that's the Fick's principle. PAH enters the kidney through the renal artery, and there are two ways a substance could leave the kidney. either by leaving through the renal vein or by going into the urine. What's unique about PAH is that at low PAH concentrations, below the transport maximum, some PAH gets filtered into the glomerulus, and the remainder of the PAH that bypasses the glomerulus gets secreted from the paratubular capillaries into the tubules. So in the end, all blood plasma leaving the kidneys is free from PAH. In other words, the amount of PAH in the renal vein is practically zero, and the amount of PAH entering the kidney via the renal artery equals the amount of PAH excreted in the urine. Now, the amount of PAH entering the kidneys is equal to the concentration of PAH in the renal artery times the renal plasma flow, or RPF for short, which is the volume of plasma delivered to the kidneys each minute. In the same way, the amount of PAH that gets excreted in the urine is equal to the concentration of PAH in the urine times the urine flow rate, which is the volume of urine formed per minute. Remember, since the amounts of PAH entering and leaving are the same, that means that these two equations are equal. We can solve for renal plasma flow by dividing by the concentration of PAH in the renal artery. So now we have the concentration of PAH in the urine, which is in milligrams of PAH per milliliter of urine, times urine flow rate, or milliliters of urine formed per minute, all divided by the concentration of PAH in the renal artery, which is also milligrams of PAH per milliliter. And when it's all said and done, you end up with renal plasma flow in milliliters of plasma per minute. And this makes it pretty easy to calculate renal plasma flow, but it's not 100% accurate, since it's possible that a tiny amount of PAH has remained in the renal vein. So the estimated renal plasma flow is usually thought to be within 10% of the true renal plasma flow. Now, to convert the renal plasma flow to renal blood flow, we also have to take into account the volume of blood occupied by red blood cells. As an example, a hematocrit of 40% means that the red blood cells occupy 40% of the blood volume, and plasma accounts for the remaining 60%. The total renal blood flow is measured by dividing the clearance of PAH by the fractional blood volume occupied by plasma, which is 1 minus the hematocrit. Normally, because all of the PAH gets cleared by the kidney, renal plasma flow equals PAH clearance. At high PAH concentrations, above the transport maximum, PAH continues to get freely filtered, but the PAH that bypasses the glomerulus starts to overwhelm the carrier proteins in the tubule. As a result, secretion will reach its saturation point, and some of this PAH will be left behind in the plasma, and PAH in the renal artery will no longer be equal to the PAH in the urine. In fact, this means that PAH in the urine starts to fall below the PAH in the renal artery. Alright, as a quick recap, below the transport maximum, all the para-aminohippuric acid, or PAH, making it to the kidneys is removed from blood that passes through the kidneys. And above the transport maximum, the carrier proteins in the nephron are saturated, and some PAH gets left in the blood. In the former case, the renal plasma flow can be estimated by taking the concentration of PAH in the urine times urine flow rate, all divided by the concentration of PAH in the renal artery.